Good day to all of our participants. I am Adrian C. Perjo, a faculty member of the Bataan Peninsula State University located in the city of Balanga in Bataan Province. My task is to communicate the journey of our research project entitled From Pest to a Valued Commodity, Black-Chin Tilapia for the Development of Surimi-Based Products. The project is about value adding of a known prolific fish farm pest in Bataan, Black-Chin Tilapia or BCT. It's an invasive species plaguing local farm ponds and deterring productivity. It's been multiplying fast and competing for feeds meant for cultured fish species. It's now been reaching growth sizes comparable to the more commercially popular Nile tilapia. It's being sold very cheaply in local public markets due to its reputation as a pest and due to its bland taste. The infestation has been a concern by the BFAR because it's been compromising the carrying capacity of local farm ponds, causing losses to fish farmers. The infestation has triggered calls to curb their population and for their productive utilization. While the species is bland tasting, it was found to be a lean and moisture-rich fish based on a preliminary study, making it a potential raw material for artificial seafood products like surimi. Originating from Japan, the Surimi industry has been spreading to many countries now. In the Philippines, the popularity of surimi-based products is apparent in the commercialization of processed fish meat delicacies, the most recognizable of which are fish balls, fish sticks, and fish cakes. Our project worked on developing the edible portion of BCT into a surimi-based product, a fish loaf, assess its quality and profitability, and then imparted the developed technology to target beneficiaries. Specifically, we characterized BCT and established its safety and quality as a food processing raw material. We developed a surimi-based product from it and evaluated its quality and stability against a leading commercial variety. We established its economic feasibility through a cost-benefit analysis. We developed processing manuals for the optimized processes, and then we imparted the developed technology to beneficiaries through capacity building activities. The project presented a means to control the BCT infestation and introduced a very cheap and abundant raw material for the fish processing industry. Through and for this project, we hope for stimulation of further innovations on developing BCT into a more valuable commodity, and of course, commercialization of our developed technology via establishment of enterprise operations and private partnerships. Also, through this project, we look forward to sustainable fishery through alternative livelihood opportunities and occupational mobility and improve fishery processes and situation. The present and following slides present the project's most salient activities, results, and outputs. The fish used in the project were sourced from BCT-infested farm ponds located in Orani, Bataan. Fish samples were caught using net traps and gill nets. Two yield indices were ascertained from the samples, processing yield and fillet yield. It was found that the processing and fillet yields of all samples were higher than the reported values for the more commercially used farm Nile tilapia. Also, it was found that the BCT samples have a comparable mineral load with Nile tilapia. The species has high moisture and protein contents too, but is very low in fat, making it a lean fish. The samples have no serious heavy metal and microbial contamination through standard tests, although appropriate post-harvest handling is still highly recommended. The physico-chemical and microbial tests established that the VCT pest can be consumed safely and has the quality as a potential raw material in surimi processing. For the surimi processing, we strictly followed international standards. We tried 8 surimi formulations using 3 common cryoprotectants, namely sucrose, sorbitol, and sodium tripolyphosphate. And based on our tests for important Surimi functional properties like water retention, foaming, emulsifiability, and gelling, the one with the best quality attributes was the 6% sucrose with 0.5% sodium tripolyphosphate formulation. Our best formulation yielded comparable figures with previous works that use more popular species for surimi processing, like lizardfish, threadfin bream, and purple-spotted big-eye. It's concluded that surimi developed from BCT, with the right formulation, can be at par in quality with those made from traditional raw materials. For the product development, the produced fish loaf exhibited a brownish color, slightly darker than that of common meat loaves. It has a firm texture, minimal crusting when cooked, and an umami flavor. So based on sensory tests, it has better acceptability than a commercial counterpart due to its more pleasant odor and taste. The product was stable up to a significant length of storage time, 4 months or more using our two types of packaging. Also, it can withstand several freeze-thaw cycles for a lengthy storage time. In terms of nutritional quality, the developed product had comparable figures with commercial fish loaves. But of course, optimizations can still be done to enhance important nutritional parameters. Additionally, results from a cost-benefit analysis of the product development suggested that the product is a profitable and feasible investment. The product can be profitable based on a 5-year cash flow projection. So all in all, it can be stated that the developed product has the potential for industrial popularization. The project was able to conduct a series of trainings on product processing for members of three cooperatives based in Orani, Bataan. The trainings were done in the form of extension work by the project team at the BPSU Fish Processing Center in BPSU Orani. The trainings were done in collaboration with the Orani Bataan local government unit. The very purpose of the trainings were to involve the community in introducing means of productively utilizing the BCT pest and to offer them additional income generating sources. Also, the project team has been able to publish scientific papers in international peer-reviewed journals. Our paper entitled Quality Attributes of Black-Chin Tilapia Surimi treated with different cryoprotectants has been approved for publication in Food Research, a Scopus Index journal based in Malaysia. It will be published next month. Another paper entitled A Baseline Study on the Quality and Safety of Consumption of a Pest Species in Bataan, Philippines, basis for its productive utilization in the fisheries sector, has been published already in the Journal of Fisheries Science, an international peer-reviewed journal based in Singapore. Presently, we are working on two more papers to be submitted for review in other journals this year. Also, we were able to present the two published papers already in two international research conferences. The project team was also able to register a full-blown processing manual for the BCT post-harvest procedures, surimi processing, and fish flow production to the National Library of the Philippines. The book bears the project title. Lastly, the project team was able to submit two utility models for registration to the Intellectual Property Office of the Philippines. Initial feedback was received by the team last November 2021. The title of the two utility models can be seen in the slide. To end this talk, our project was able to develop a product from a recognized fish farm pest with a very high potential for commercialization. We were able to do it using easily accessible and cheap raw materials and using very straightforward processing procedures. Presently, our product development will be put under our university's technology business incubation arm to prime it for commercialization. The project team hopes for accelerated and sustained adoption of our product development through partnerships with enterprises and further innovations on developing the pest into a valuable commodity. Maraming salamat po.